My name is Cindy Wu. We have the three authors of our feature paper here on our Zoom meeting, and I just want to let you know that the format of tonight's discussion will be a brief introduction by myself, and then some slides kind of putting the article into historical context, and then we have left a lot of room for some questions directly of the authors. So if you're just tuning in, if you have questions for the authors at the end, feel free to use your chat button. It's located on the lower taskbar of the Zoom meeting. If you just put in your question there, we will go through your questions and then we'll call on you, and then as you start to speak, you'll be unmuted, and then you can have the opportunity to ask the authors what your questions are. So Phaedra, if I could have my slides, please.
Okay.
So today we have our three authors, Dr. Mindy Haws and Dr. Mary Gingrass, who are both in private practice at the Plastic Surgery Center of Nashville, and Dr. Stephen Porter, who is the Chief Scientific Officer of Integrated Healing Technologies. Next slide, please. So for disclosures, Dr. Mindy Haws and Dr. Mary Gingrass are both investors in Integrated Healing Technologies, and Dr. Stephen Porter is the Chief Scientific Advisor of Integrated Healing Technologies. So as we all know, the most common breast augmentation complications are capsular contracture, hematoma, seroma, and infections. In fact, infections make up about 1.5 to 2.5% of all cosmetic breast surgeries and 25 to 35% of reconstructive breast surgeries. It is also known that bacterial biofilms are the cause of chronic infections, which are estimated to cost the US about $1 billion in revisional breast surgeries. Now recently, data has come out to implicate Ralstonia pachydae biofilm as linked to BIA-ALCL. And in a recent study of 1,900 plastic surgeons surveyed, about 50% of them report irrigating with chlorhexidine, about 50% with triple antibiotic solution, and 44% with iodine in the surgical pocket. Next slide, please. So just to put this article into context, so there was an article out of Australia by Hugh et al. In November 2018, which compared hypochlorous acid with iodine irrigation in eradicating Staph aureus from implant surfaces. And this is one of their figures. And if you look here, it shows that at 100% concentration that they, and according to their study, phase one, previous slide.
Sorry about that, my bad.
Okay, that phase one in the presence of bovine serum was ineffective at eradicating Staph aureus. And they use this to say that phase one is not effective, was not effective compared to betadine in eradicating Staph aureus. However, the study brought up some concerns, next slide, namely, which were addressed by Dr. Jack Fisher's article, in that they, in vitro conditions under which they studied HOCl was not clinically relevant, and namely that the 5% BCS, the bovine calf serum concentration, was too high, which effectively inactivated all the HOCl binding sites, which is not clinically relevant. And so this led the authors of the current study to ask some of these questions that were clinically relevant, and specifically, what is the effective concentration of hypochlorous acid? What is the protein concentration in the pocket after adequate surgical hemostasis? And then lastly, what is the effective concentration of hypochlorous acid available after protein binding? And this really called for a need for an in vivo study to answer all of these questions. And this is where we are discussing the results of this article today. So with that, I'll turn that over to Dr. Haws to discuss their work.
Thank you, Dr. Wu. As Dr. Wu brought up, intuitively we felt like the fluid that we see in pocket after dissection was not high in protein, because after all, we don't want to see any fluid in the pocket when we put the implant in. But nobody had that data. So our study was designed, we used 18 consecutive subglandular and subpectoral breast augmentations, primaries. After we achieved hemostasis of a new pocket, we irrigated the pocket with 20 cc's of saline, left it in the pocket for a minute, and then collected the fluid. That was then measured for albumin and total protein. We also, after we irrigated with the saline, then placed phase 1 within the pocket. This was allowed to sit for 1 minute, and we collected it and sent it also for evaluation. What we found was the amount of protein in the pocket was reflective of interstitial protein numbers, not intravascular serum numbers, which is very low compared to serum numbers. And that, in fact, given the amount of protein that was in the pocket, there was plenty of HOCl available to bind and not be taken up by the protein and therefore inactivated. So our conclusion was phase 1 irrigation, there is more than sufficient HOCl molecules for antimicrobial, antibiofilm activity because of the interstitial fluid component of the pocket.
So basically, our paper refutes the in vitro study by showing that in an in vivo setting, a normal everyday breast augmentation pocket, that the protein content is nowhere near what they were using with this bovine calf serum. So the in vitro study really has no relevance to the clinical in vivo study. And so, you know, in a normal everyday setting, the protein content really hadn't been documented. It wasn't out there in the literature. And so that was really the point of this study. And I think that at this point, Dr. Porter is going to add a little bit to our knowledge about you know, some of the other benefits. This is a totally non-toxic solution. It's an excellent antimicrobial. It's excellent antibiofilm. It has a lot of potential benefits and very, very low to little risk to the patient. So Dr. Haws and I have been using it in a clinical setting for some time now and have, you know, had excellent experience with it. Dr. Porter, do you want to make a comment?
Yeah, is there a slide for me to speak from, or I can just do the, no, okay, fine. So from my perspective, and the purpose for us collaborating with the surgical team is that we wanted to document in a real world setting an in vivo model in which we would document the amount of protein that would be intrinsically available during the surgical procedure, and then theoretically what would bind all or any of the HOCl. And it turns out there's more than enough, as we have espoused. And I'd just like to embellish from my position as a scientific advisor, and not as somebody espousing any off-label claims, but basically fundamentally to explain what we consider possible extracurricular activities and benefits of HOCl in these procedures. Of course, HOCl is a highly reactive oxidative process that will bind to virtually any peptide, protein, cytokine, inflammatory cytokine, proteinaceous material, including red cells, white cells, that are in that pocket. And those inflammatory processes are tamped down, if not completely neutralized, by the presence of HOCl. We've demonstrated in other studies the complete neutralization of toxins, exotoxins and endotoxins. About 14 different toxins have been completely neutralized by HOCl, which turns out to be extremely beneficial in neck fash patients, excuse me. The other beneficial effect is diminishing or minimizing, because of the anti-inflammatory effects, the additional capillary leak and possible hematoma and seroma formation. And with additional studies, I suggest that we perhaps do some more quantitative assessment of post-operative healing times, seroma and hematoma formation or presence, and see if that is substantiated by the theoretical benefit of HOCl. The other thing, which I think should be speaking to the safety of this product, is once it is bound either to biofilm in multiple sites, bacteria, fungi, viruses, or cytokines, or present interstitial albumin or immunoglobulins, the remaining components of a phase 1 solution is saline and water. So thus, you can retain the product in the pocket without having to do additional irrigation for additional time savings in the procedure, but also the standpoint that you don't have a retained toxic molecule, potentially toxic cytotoxic molecule in the breast pocket. And I think that's the essence of what I wanted to say. I can answer certainly more questions.
Dr. Porter, I think the other really important thing to know that you had mentioned to us a little earlier was that the half-life of phase 1 is short, right? And I think that's important to point out to the listeners because that would be important for people to know.
Well, there's the distinction of terms, half-life versus effective pharmacologic activity versus time to kill. So time kill studies show 20 plus number of bacteria have been neutralized by HOCl or inactivated or killed in less than a minute. And again, the rapidity of that onset is much, much greater than virtually any antibiotic, commercial antibiotic out there. So very rapid acting, but therefore also very rapidly consumed, and therefore no additional long-term cytotoxicity should be present unless there's a vast quantity left in place, which we don't do.
So the onset's quick. So in terms of questions, should we turn it over to some questions now from the audience?
Sure, we can do that. I also wanted to point out that Dr. Van Natta is on the line, and I know he had written a commentary in the journal. So I don't know, Dr. Van Natta, or do you have any thoughts or comments at this point? Sorry to put you on the spot.
No, that's fine. Let's see.
Whoops.
Oh, we've muted him.
Let's see, I'm unmuting him. I did, and he muted himself. Hang on one second. We gotta find him.
Okay, how are we now?
Perfect.
We can hear you.
All right, sorry about that. This is, I think, an incredibly important paper, and my comments in the commentary were directed at the importance of this. The study by Dr. Hugh, who we have to acknowledge is a well-respected researcher, but she's not a clinician, and their analogy was that this would be the BCS that they used would be the equivalent. I believe they said it was either 5 or 10 cc's of blood in the pocket, which Mindy, I think you indicated, that would indicate that we have a problem as clinicians. We don't want to see that amount of blood. We want a dry pocket. And so I think it's important in this day and age of ALCL, not to mention capsular contracture, but we know there are documented cases in which betadine was used properly, allowing a 2-minute dwell time in the pocket where we still had the development of BIA ALCL. So I think it's important to be on the lookout for other options. And I think I didn't mention this, but I don't have any financial involvement with phase 1 at all. I'm completely independent on this. I believe it's important to have more tools in our toolbox, and I think this is an exciting and important one for all the reasons that have been cited. There just isn't an issue of toxicity. It kills virtually everything that we would be concerned about, and does it in a very effective manner, not to mention the disruption of biofilm. So when we have cases of capsular contracture, where we know there's a great chance of biofilms being present, when we do a capsulectomy, I think this is a tremendous adjunct in those cases to perhaps eliminate residual biofilm and again, as Dr. Porter mentioned, we need to have some more studies where we can show perhaps a reduced incidence of recurrent capsular contracture by using HOCl. I would just like to finish and ask a quick question of Mindy and Mary. You mentioned, Mindy, I believe that you washed or you placed 20 cc's of saline in the pocket. Could you clarify? I'm not sure from your paper if this was stated. Did you guys irrigate those pockets after completing the dissection, as most of us do clinically, with a few asepto syringes of saline prior to that measurement, or was this an undiluted specimen?
This was an undiluted specimen, unless we got into such extreme bleeding, you had to irrigate a little bit. But our plan was to dissect the pocket, have a dry pocket, and then irrigate with the 20 cc's of saline to see what was there without diluting it further with more saline.
Okay, and I think that's really important to take into consideration, because we know clinically, virtually all of us, I would say it's almost the standard of care, or after we complete a dissection, would irrigate that pocket. So the measurement you guys took with that really would be a maximum.
Sure.
Yeah, it should be high.
That's exactly right. So,
okay, and that's an important distinction.
Yep. And thus, you're in for a free to conclusion would be there being an even more active HOCl.
Absolutely.
If that's the case. That's right.
Absolutely.
I can also comment in how well this is tolerated, because I've used this solution in some patients under local anesthesia, and I was kind of nervous about it at first. Like, is it going to sting? Is it going to, burn? It's an acid after all, and it's extremely well tolerated. Another clinical observation that Dr. Haws and I had was that it almost seems a little hemostatic, like it. When you'd irrigate the pocket, it almost seemed like any little tiny bit of oozing or something that might be going on would always be finished up by the time you open the pocket back up. So that would be interesting to see, is it really doing that? And theoretically, that would be decreasing the capillary leak.
Yeah, that's exactly the case. But even people have described some slight blanching. The opposite is true with some veterinary experiences, where they use a microsim, which has bleach in it, and you get a hyperemia. You get a really red, serious, irritated-looking pocket in those surgical procedures with horses that we've experienced.
Well, we're getting some questions that are starting to roll in, and I believe
Okay.
Dr. Molliver had a question. So if Dr. Molliver, you wanted to ask your question?
Sure. First of all, let me qualify what I'm about to say by saying, good study. I think we really obviously need to look at biofilm and capsule contracture and what we can do to eradicate bacteria and all forms of bacteria. But I have to say, I'm right off the bat a little bit concerned. We have two investors and one of the people from the company that are presenting the paper. Now, on the one hand, who else is going to present it? Because they know about it and we don't. So it's a good paper, and I applaud it, but how do you respond to the rest of us that say, well, wait a second, there's inherent bias right from the get-go in these papers, and some might even say, maybe question the validity. I'm a little concerned.
I think anybody who's doing primary breast augmentation could repeat this study and get exactly the same results. I mean, and also IHT is actually no longer in existence, so I have no financial interest in this product whatsoever.
Oh, bravo! I like it even better.
The company's
I'm sorry.
Everything we're doing now is gratis. Just we would like to see phase 1 go on, but we currently have no financial interest except for the money we lost.
We injected in Dr. Fisher's in his wound vac, which was a great vac, but.
All right. Well, I'm glad to hear that. I'd love to see the journal look out.
Actually,
I'm sorry?
It works out in our favor, I think, for legitimacy.
I think so too. I like to hear that. That's a good comeback. I like that.
Thank you, Dr. Molliver, for your question. There's also another question from the audience from Miriam regarding the effect, if any, on implant integrity of irrigating with phase 1, whether or not we know if it has any effect at all onto a smooth or textured implant surface.
Stephen can answer this specifically. Now, this is all FDA off-label use, but he can talk. This was tried in the lab.
We specifically measured a number of breast implants for biocompatibility, tensile strength, and also any degradation of the product, and also any leaching of the products of compounds of interest. And those studies were done and conducted under a GLP laboratory, independent laboratory in California. And I would be glad to share any of those results with people of interest. To summarize, there is no effect on the tensile strength. I mean, the concern would be that a solution would weaken the implant, it would leak and break. The other conclusion would be that it would leach something toxic or onerous and produce serious inflammatory side effects, and in both cases, nothing occurred. I will also tell you that we tested the Kellner funnel, and that's also found to be totally innocuous under the presence of up to 24 hours exposure, which is clinically irrelevant, when no one would soak their implant for 24 hours prior to implantation, and no one would use a colored funnel for 24 hours before using it during surgical procedures. So anyway, you have an innocuous compound that does not interfere with the implant materials in any way.
Steve, will that be published?
I'm sure. I have no problem getting it published. I think it would be great if the journal would welcome the publication. I'd be glad. It's scientifically rigorously done and conducted. I think it's an important piece to add to the literature. And I'll say the reason why is you all may be aware, for many years, iodine, betadine, has come under extreme scrutiny about its possible effects on implants, and was black boxed and not allowed for almost a decade or more because of a study that was done with catheters in the presence of betadine. And then in 2010 or 12, it was allowed to be reused again, but it still requires in labeling that betadine has to be flushed from the pocket if it is used in breast implants. Again, because of material interactions, they were concerned with the iodine.
This also basically becomes normal saline in 1 minute. So, you're preparing your implant, it's neutralized by the time you put your implant in.
Yep.
And could I offer a completely unscientific comment that according to Jack Fisher, you can drink this stuff. Now, we all know we can't trust a lot of what Jack says, but I will admit the fact that I took the little spray bottle of this and I've sprayed it in my mouth a couple of times, and I still have all my taste buds. So I'd just like to point out, I think there's a margin of safety here.
Yeah, you can do that. You can gargle with it if you have a sore throat. You can spray it up your nose for sinusitis. You can put it in your eyes for EKC. I mean, it is really a very, very helpful product. You can use it for bee stings and spider bites and many, many other inflammatory, histamine-related or toxin-related.
Because it has an anti-inflammatory component.
And both anti-inflammatory, but neutralizing of the toxins and the histamines and the cytokines. So you get almost an immediate effect from the use of this product.
Well, thank you. That's a great segue for Dr. Fisher, who is also on the call, and he wrote the commentary on Dr. Hughes' paper. And forgive me for calling you out, but I just wanted to see if you had any comments on this paper or just any of the discussion.
So, can you hear me?
Yes.
Okay. I'm always intimidated by technology. So last year we actually, I had a wonderful experience of spending the holidays last year giving enemas to pigs with hypochlorous. Live till you've given a pig an enema. But what we demonstrated...
I was there too, by the way.
Yeah, you were there. Yes, Steve was there too. Now, the other thing I want to point out, all these studies were done by independent labs. So I understand Clayton's concern But remember, everything was sent out to a third party for analysis. So what was returned was very, very independently controlled. But we did do the enemas to see what would happen with it present in the colon for an hour. And not surprisingly, all the data came back showing it had absolutely no effect on the mucosa. This particular product we're using was actually developed for ophthalmology in a slightly weaker strength. So that's one of the things that attracted us to it, is the data and all the research that had been done with ophthalmology.
Well, let me look to the other questions. I actually had a question, if there's no one else that had one right now. It's for the authors. You know, in the conclusion, you allude to future studies are, I think the wording is, future studies are underway to demonstrate the safety of HOCl and textured and smooth implants. Now, are you allowed to?
We already did that study. We did that study. It's an in vitro study. It's not an in vivo, but basically we looked at all. Jack, you have to help me how many varieties we had. We had 3 or 4 of the leading breast implant materials that we actually did an extensive long-term evaluation of the compatibility of those products in the presence of HOCl. It would be the precursor for asking for an additional FDA labeling claim, and it would be a required study by them to show biocompatibility.
But can you still hear me?
Yes.
Yeah, that's off-label use. In other words, we have the data, but like a lot of things that we use, until you formally submit it, we still have to clarify that that is off-label use. But as we all know, a lot that we've done in our careers is off-label. But we have to be very careful about making any specific claims at this time. I see Dr. Jewell's face.
Hi.
Hi, Mark.
Good evening.
I also saw, didn't Dr. Kenkel try to ask a question earlier?
Is he still on, Dr. Kinkel?
Does Dr. Jewell have a question?
Was it interrupt?
Yeah, Dr. Kenkel.
I'm here. I'm just listening and learning.
As we all are.
For anybody else who's interested in clinical relevance, Dr. Haws and I have been using this, you know, for any wounds, and we have these little spray bottles, and you can send it home with the patient. And I can't tell you how much easier just little plastic surgery wound care is with this product compared to Dakin solution and all this stuff. I haven't seen a Pseudomonas, a green dressing, come out in a year and a half at my office. Like it really is pretty good stuff.
That's great. So for people who are interested in getting it, you guys have mentioned before that IHT is no longer distributing phase 1. I'm assuming that it's being distributed through another company, that it's still available?
Yeah, so it's in receivership, but they want to sell the stuff. There is more than enough product to last a couple years. The other thing that wasn't mentioned is it has a 3-year shelf life, and it has a wide range of tolerance of temperatures. So that's very important. But it's readily available and can be easily ordered. It's just those of us are out of the loop with that. But I don't want to do any advertising on a journal club, but if anybody is interested, they can email Mindy, Mary, or me, and we can at least refer you to the distributor. Remember, what went bankrupt was the distributor. The company's still manufacturing the product. So it's not like the company is out. It's still there. And again, to make Clayton feel better, we've all lost money, Clayton, and we're all
study
But
Can't keep
I
doing
second that.
yeah, but I feel so strongly about this product, and we have salvaged so many difficult cases that even though I have no relationship with it, I'm going to continue to talk about it and help promote it because I really have a lot of confidence in its efficacy. Nothing we have is a miracle, but I think this product, especially with biofilm, is quite unique.
Dr. Jewell, I think that's a perfect segue to your question.
Right. My question was, what is the cost of this product?
Our cost, I believe, is about $50 per patient, and that's $50 for like 235 cc bottle.
Okay. Thank you.
I think Dr. Wickstrom wanted to say something, and he's smarter than the rest of us.
I can attest to that.
I don't know about that, but anyhow, yeah, I just had one comment to add. I wrote a commentary to the Hugh et al. Article, and I think there's a broader message from this study and the study that it was designed to address. And that is with a lot of the biofilm testing, there's been more of that published lately with different breast implant surfaces. And although they are using, in many of these cases, a standard biofilm test method, such as the CDC bioreactor or some of the other ones, they're using standard test conditions. And if you look at other testing in other areas of biofilm research, they oftentimes adapt those conditions to make them more relevant. At a biofilm conference earlier this year, they were doing studies in the dairy industry where you have milk with very high levels of protein, much more nutrients available, and they modified the conditions. So I think this study really illustrates that for any study involving breast implant surfaces and biofilms, one really should make sure that the conditions being used are reflective of the actual clinical situation, not just default standard test methods.
Yeah, and again, let me embellish. The Hugh paper was based on the TGA, the Australian equivalent of our FDA, and it was a standardized test for an antiseptic, and they used this. The protein is called a soil test, and it clearly wasn't relevant in this particular case, in my opinion.
I believe we have another question from Dr. Zamani.
Thank you, Cindy. Can you hear me well?
Yes.
Okay, perfect. So the questions I have actually are quite relevant to what we're discussing right now. It's about whether any of the surgeons who are quite familiar with phase 1 solution have had the opportunity to examine any implants that have had to be removed after having been inserted during that initial surgery, having used phase 1, and whether they've had the opportunity to examine the surface of the implant for biofilm and compare it to, you know, what you would see with other irrigation methods, is the first part of my question. Secondly, whether in your clinical practice after adopting phase 1 solutions, you've noticed any change in sort of problems that can arise after primary breast augmentation, such as decrease in your capsular contracture rates, infections, implant failures, or anything of the such?
We haven't been using it long enough, I think, to tell long-term complications like capsular contracture. And I have to admit, I'm sure I have gone back for one reason or another to do an implant exchange, and certainly didn't notice anything grossly, but that's a great idea to study those implants for biofilms. But no, I haven't specifically noticed any difference in, and I would say post-op, like seroma isn't really a big problem with early breast augmentation, patients. So I haven't noticed any difference in that.
If I could chime in, am I still part of this?
Mm-hmm.
As far as
Yeah. So a paper was just submitted by Dr. Josh Halprin. He took 15 Baker 3s and 4s and all he did was do a capsulectomy, put a new implant in, but most importantly, put it in the same pocket. His average follow-up now is at about a year, and he just submitted a paper to ASAPS on it, and so far they're all Baker 1. Now, as you know, previous studies have shown that you need to follow a little more than about a year. 50% of capsules tend to recur in about a year, but if you're following out to about a year and a half to 2 years, you're probably reaching at least 80 to 85% of the capsules. But what I find interesting about his results is, as we all know clinically, these people who've already had a capsule, they tend to recur very quickly. So this is a very promising number. It's small, but remember, each one of those patients is their own control.
I think it's also important, Jack, to tell people that if you're putting it in specifically for biofilm, like if you're doing a capsulectomy and an implant exchange, you need to keep it in for 5 minutes as opposed to the 1 minute.
Correct.
So I just wanted to do a last call for questions. I think all of us are unmuted, so if we have any other questions, feel free to ask them now.
Cindy, I'd just like to offer, just to kind of add to what Mary just said, and I'd be interested in how others are using it, but when I place the hypochlorous in, I place some in the pocket. After irrigating the pocket, I leave some in the pocket, place the implant and close with it in place. So that dwell time technically ends up being much longer, maybe than even 5 minutes. And of course, as we've already stated, it's going to convert into water ultimately and be resorbed.
Saline.
I would agree with that, and also, as I said, the Keller funnel works. The whole process, 4 steps for wetting the Keller funnel, and you could even use HOCl during that process.
Tell us again how long it takes in your experience before it converts to water.
It's seconds, minutes. As soon as it touches the protein, peptide, any surface that's going to neutralize, the highly oxidative reactive process that binds to disulfhydryl bonds. Basically, technically, any protein peptide molecule is going to be instantly affected by the HOCl. In the case of bacteria, they do kill times, and they're measuring in 10 seconds the kill times.
So the whole dwell time of 2 minutes versus 5 minutes becomes moot point.
Well, it just depends on how much extra HOCl, it depends on, again, what we've just demonstrated, how much starting protein is there.
Okay.
And so that's a huge variable, and you don't know. So if you've got more dwell time, you're just going to get even more protein neutralized.
Yeah, let me add to that. The reason we came up with 5 minutes for a recurrent case is that's when we studied the biofilm formed on the implants.
Right.
So it probably is quicker than that, but we don't want to shortchange it based on our own data. What's unique about HOCl is it has no electrical charge, unlike other products. And because it has no charge, that's what allows it to penetrate biofilm very quickly. If you use something like sodium hypochlorite, which is bleach, the problem is it has an anionic charge, and so it has trouble, if not impossible, to penetrate a biofilm. So HOCl is highly effective, and I think that's why it's had good results in dealing with infections with biofilm, because it so readily penetrates the biofilm, and that's basic chemistry.
Well, it also, to segue on that, Jack, it also, because of its neutrality, it readily penetrates spores. So in the case of C. diff, you can get the vegetative form, but you're hard to kill the spore. Well, HOCl kills the spores by being able to pass into the pores of the spore, as in the case of the quote pores or aqueous channels of biofilm.
Chlorhexidine is very good at getting and killing planktonic bacteria. So is betadine. Planktonic just means free floating.
Right.
The difference is chlorhexidine cannot really penetrate biofilm. And again, that's basic chemistry. The biguanides have trouble penetrating anything like the mucopolysaccharide that makes up a biofilm. And if I sound really smart,
that's really good, Jack. I got to give you.
Yeah, if I sound really smart, that's because Roger Wicks and Steve Porter have taught me all this.
This has been a very stimulating conversation. I want to make one last call for any questions or comments from people. We want to be mindful of people's time. I think we could keep going, but we have people in different time zones.
This is a wonderful format.
Yeah.
I was very hesitant about this, especially since anything with a computer scares me. But I have to tell you, I'm very impressed with this. And I think it's wonderful that ASJ is doing this.
Yeah, kudos to them. I agree.
Yeah. Well, thank you to Phaedra and Hunter for facilitating all this, and also Dr. Tatini for starting off the Journal Club with this format. I think this is a wonderful format, and I think we'll continue this way. So with that, any other last comments or questions for our speakers? I think it's been a great discussion.
It's been great. Thank you for having all of us.
Well, thank you to
This is wonderful. Very innovative. Thanks.